Welcome, class, to your second lecture of Chapter 11, Lipids and Membranes, as part of our biochemistry course. This lecture shouldn't take too long, so hope that you have your notebooks out, ready to take some notes, and perhaps even your student notes, ready to fill in some of these blanks. All right, so here's our chapter theme from Ralph Waldo Emerson. All that I have seen teaches me to trust the Creator for all I have not seen. So keep in mind, y'all, we are studying biomolecules, and it seems like we're actually studying a factory or some sort of site logistics, but really this is going on in your body all the time because this is the way God created it. So I hope that as you study this material, you are glorifying God through creation. So first I'm going to talk about what lipoproteins are. So lipoproteins are these large structures. You can see it right here. It's a term most often applied to molecular complexes found in the blood plasma of mammals. Blood plasma. And they serve as transport lipid molecules. Transport lipid molecules through the bloodstream from organ to organ. The protein components, which are called apolipoproteins, for lipoproteins, they're synthesized in the liver or the intestines. So I'm going to unpack what I just talked about right here as we look at this drawing over here on the left side of this slide. So lipoproteins, they tend to have this sort of structure, and what you'll notice indicated in blue is cholesterol. Okay, remember the steroid skeleton that cholesterol has? I'm not going to actually draw cholesterol, but I will draw the steroid ring, a skeleton. That's 666, and then a 5-membered ring right here. And so that is cholesterol. Recall that cholesterol serves in membranes as structural components. And here they may serve as the same thing, but then as you look at these little purple molecules, those are going to be your fatty acids, right? Your phospholipids. And together they make up this ball-like structure. And you'll see these kind of colored noodle sections around here. And of course, here's a little cross section right here. There actually is, that noodle would connect all the way through, but it's trying to show you what's going on inside of this lipoprotein. But these are proteins that serve as, they interact with receptors in the place that they're going to go. So keep in mind that this lipoprotein as a whole, if you were to take off that protein component, meaning look at this drawing without those little noodles around it, it wouldn't know where to go because there would be nowhere to receive it. So there are these proteins on it that are specific for whatever type of lipoprotein it is to govern which type of cell or tissue it's going to go to. And so it's going to meet with a receptor, and that receptor is going to accept what this has to give. Okay, so these are transport molecules. Like I said, when you think of transport molecule, just think of like cross-country drivers or those truckers, right? They are carrying goods from one place to another, and that's exactly what these things do. So what are they carrying? Well, they're carrying lipid molecules. They're carrying lipid molecules. It can be fatty acids or triacylglycerides, which are triacylglycerides, excuse me, or it can be cholesterol. And your fatty acids and your cholesterol, they're going to find their way in the middle portion of this structure, okay? So they're not going to be bound to the outside. They're going to be inside this ball-like structure as the integrity that surrounds it is going to enclose it, okay? And as I said right here in this third bullet point, the protein components, which are called apolipoproteins, what I'm talking about there is the noodle-looking structures. Those are your apolipoproteins. They're the protein components that interact with the receptors of which these are going to, okay? And like I said, those components, which by themselves do not make a lipoprotein, but when connected or bound to the entire structure, which has your cholesterol and phospholipids, it makes a lipoprotein. But the noodle-like structures, aka the apolipoproteins, they are made or synthesized in the liver or the intestines, okay? So I just wanted you to get wind of that. So there's many components going on here. There's a function that's going on, and there's also receiver. Just like whenever a truck driver drives across the country and it enters into, say, UPS, there's going to be some checkpoint to verify that that truck belongs in that location. That is what those protein components are doing, okay? They get you through the gate, if you will. So in the previous slide, I introduced what lipoproteins are. Now I'm going to introduce to you the 4 classifications of lipoproteins. First on the list are chylomicrons. These are very interesting lipoproteins. They're very large, and they're of extremely low density. And what that means is that they don't have a large protein component to them. They're made up of other things that are low density, and what they do is they transport triacylglycerol and cholesterol esters, which are synthesized in the intestines. Okay, so chylomicrons, you can think of this. They are the ones that takes dietary lipids. Okay, and so you eat some food, it breaks up into proteins, carbohydrates, and lipids. Okay, and lipids can be cholesterol and fatty acids. We've already learned that. And so what chylomicrons do is once it goes to the stomach and then your intestines, these chylomicrons pick up the lipids left over, which are triacylglycerols, fatty acids, and cholesterol esters, and then it sends them or transports them to either your tissues, the individual cells, or it can go to the intestines. Sorry, not the intestines, the liver. It goes to the liver. And that's what they do. Once they're done giving off their cholesterol esters and the triacylglycerols, they then go to the liver to be degraded and to be done with. Next up on the list, number 2, you have VLDLs, very low density lipoproteins. And what these do is they are synthesized in the liver and they transport lipids to the tissues. Now, when I say lipids right here, lipids, what I really mean is TGs, triacylglycerols, fatty acids, those sort of things. They don't really transport cholesterol. We'll talk a little bit about what does transport cholesterol, but imagine this. You just eat your food, your chylomicrons bring, cholesterol and TGs to your other cells, tissues, your liver, and these chylomicrons are synthesized in the intestines. And now you have your bodily triacylglycerols and cholesterol esters. Now you have these next 3 lipoproteins that they transport lipids, but not from the dietary source. Your chylomicrons did that. Now that your lipids are in the places that they're supposed to be, then these other 3 come in and transport them from their current location to wherever your body needs them. Okay, so what happens is fats, we're going to learn in the next chapter, really give off a lot of energy through beta oxidation and other processes. And so when your body needs some energy, it's going to start to burn fat for energy. And so what happens is your VLDLs will first transport these lipids, and really when I say lipids, your triacylglycerides or your fatty acids, to tissues in order to be used. Now, once it dispenses off, now keep in mind these also have cholesterol esters, cholesterol on them, but they're not dispensing the cholesterol, they're dispensing the triacylglycerides. And once they do that, they go through an intermediate called IDLs, and then it ends up going to LDLs, okay? And so very low density lipoprotein, after going through an intermediate, that intermediate being after it gets rid of the triacylglyceride, then goes to an IDL, and that IDL eventually turns into a low density lipoprotein, LDLs. Now, low density lipoproteins are interesting as well. They're the principal transporter of cholesterol. And so, whereas the VLDLs transport the triacylglycerides or fatty acids, but as they dispense those off, they gain some density to go from very low density to low density, but they retain the cholesterol aspect of them. They go through that IDL intermediate, then become LDLs, and then LDLs then give their cholesterol to your tissues and whatnot. And then once it gets done getting rid of all those, your cholesterol and your triacylglycerides or TGs, it then gets degraded in the liver and then it's able to be disposed of. Lastly, our final one is called HDL. Those are high-density lipoproteins, and it's a protein-rich particle because protein-rich means that it is high density. Proteins are very dense. So that's how you can think about these. Chylomicrons have a very low protein component. High-density lipoproteins have a very high protein component. These are produced in the liver and intestines, and they seem to be a scavenger of excess cholesterol from membranes. So you can think of these as good cholesterol. So imagine this. You have your chylomicrons giving off the lipids from dietary sources, then your VLDL gives off the TGs, and then your LDLs give off the cholesterol. And now your body has all this stuff, and if your body doesn't burn it, you have a lot of cholesterol buildup and a lot of fat buildup. Well, of course it can turn into fatty tissue, but also if there's excess cholesterol floating around, these high-density lipoproteins go around and scavenge these extra cholesterol and fatty acids, in which you can think about these as almost like a sponge, okay? It absorbs it, and then gets rid of it. And it does not absorb the fatty acids. It just absorbs cholesterol. Excuse me on that one. But you can read more about these in your book, because in the next slide we're going to see the actual pie charts of the ratios of the components of each of these lipoproteins. But I just wanted to let you know that each of these has a purpose. But if we can increase our amount of HDLs and decrease the amount of VLDLs, you have a pretty good diet. And there's ways to do it. You can read about that in your book whenever you get a chance. So here are the various lipoprotein compositions. So here's chylomicron. Remember, this is extremely low density. There's that glitch, class. Don't you love it? Okay. And then whenever you get to HDL, you see high density. Okay, so notice this has 55% protein component. And this one only has about 2% protein component, chylomicron. So remember, this one is from your diet. And then the other three take the already existing cholesterol and fats and transport them where they need to go. All right, and keep in mind chylomicrons are 85% triacylglycerols, which are fatty acids and whatnot. And then cholesterol ester, you can think of those as cholesterol. It's only 3% that. Whenever you go to LDLs, notice you bump up to 38% cholesterol ester. And really what I told you is that VLDLs end up going to LDLs. And now what you may notice is that VLDLs have 50% triacylglycerols. That's because its main purpose is to dispense TGs to other tissues and the liver. Once it gets rid of those, you now notice a lower percentage of those TGs, and now you see a higher percentage of cholesterol going from 12% to 38%. Okay, so don't forget it goes through that IDL intermediate, and then you form the LDL. The LDL's chief purpose is to dispense cholesterol esters to tissues. Okay, and that makes sense. The cholesterol is the highest portion of LDLs, whereas in VLDLs the highest portion is TGs. Okay, and then finally you have your HDLs, which are 55% protein component, 24% phospholipid, a mere 15% cholesterol, and you only have 4% of TGs. So don't memorize the numbers, class, but have a sense of what the components are of each of these four classes of lipoproteins. All right, class, now we're going to move on to membranes. So what is a membrane? A membrane is a non-covalent heteropolymer of lipid bilayer and associated proteins called the fluid mosaic model. Maybe you've heard that name before. Maybe you haven't. That's what we call the phospholipid bilayer. So let's break down what each of these words mean. Non-covalent just means it doesn't have the covalent bonds we know and love between two nonmetals. And so they're just interacting closely, likely through intermolecular forces or interactions. Hetero, meaning different. Polymer, meaning repeating units. Lipid bilayer, remember the phospholipid bilayer, where you have the fatty acid tails, the polar heads, containing the phosphate group, the long hydrocarbon tails that are aliphatic and hydrophobic, meaning they don't interact well with water. And they spontaneously come together. And so whenever you look at membrane structure, and there's also associated proteins, glycoproteins and whatnot, and proportions of the lipid, protein, and the carbohydrate components, carbohydrates, vary greatly between cells. Okay, there's a considerable difference between cell types and organelles. So some membranes might have high carbohydrate content, like glycolipids, glycoproteins, and whatnot. Others might have a higher lipid content, very few proteins, very few glycolipids. And keep in mind the lipids would be the phospholipids and cholesterol, okay? Cholesterol for structural integrity, and the phospholipids to maintain fluidity. And that's more fatty acid derived, and the other one isn't. And so that's how you can distinguish between cells. If there's like a large protein or carbohydrate component, it might differ from other cells that have a high lipid constituents. So I encourage y'all to watch this video right here. It's going to be helpful when studying these membranes. I want to keep in mind that these are fluid, and you know, cells are living things, and there's a lot of transactions going on, a lot of communication that needs to happen. So if you have molecule X on this side that needs to get over here. You might be able to flip-flop that using a flipase. You can read about that in your book. It goes from that side from this side to get inside the cell, let's say, to achieve its function. You have rotation where it kind of is spinning around like a merry-go-round, and so that provides movement. And then, of course, this lateral diffusion where it stays on the same side of the membrane, but it can like cause this triangle to be pushed that direction by lateral movement. Okay, again, your book talks about that, and feel free to read more about lateral diffusion, for these can all show up on the test, and I do expect you to read and do your best to comprehend these. And sure, you can look up YouTube videos as well and get further clarification on this subject matter. So when talking about what a membrane is, we got to consider membrane lipids. Membrane lipids, and they are phospholipids that form bilayers. We talked about the phospholipid bilayer already, and this happens at relatively low concentration. You don't need a lot of it to have a phospholipid bilayer spontaneously form. This is the basis of membrane structure. When we think of membranes, you think of a phospholipid bilayer. And remember, phospholipid is a type of lipid. Remember, it has that glycerol backbone, and it has a phosphate group and 2 fatty acids attached. So go back to last lecture if you need some extra help remembering what that is. Membrane lipids are largely responsible for many of the membrane properties. For instance, membrane fluidity is something we have to discuss, and it refers to the viscosity of the lipid bilayer, the viscosity of the membrane bilayer. Also, as I talked about in the last slide, there's rapid lateral movement, and it's apparently responsible for normal membrane function. See this lateral diffusion right there? Things can go to the left or the right, but notice it's staying on the same side of the lipid bilayer. It's responsible for normal membrane function. Okay? The membrane's rigid structure is due to cholesterol being embedded within it. Keep in mind, cholesterol is a lipid, and so when talking about a membrane, you have phospholipids, but you also have cholesterol buried in there. And then next you have the membrane's fluidity, discussed right here. Viscosity, it's due to the hydrocarbon tail, the hydrophobic portion. Okay, so you might want to look at some of the videos to see what a membrane actually looks like. It's really hard to show in just like a stationary slide like I have right here, but I just wanted to let you know that these are not stationary structures and that they are involved in really a lot in cell signaling, cell communication, and cell structure, and cell receptors, and what goes in a cell and what goes out of a cell, how molecules are diffused across or even flip-flopped across the membrane from one side to another using flipases. So there's a lot going on with these bilayers, and they spontaneously form even at low concentrations, and lipids are the primary constituent of these bilayers. So in terms of qualities of membranes, we got to discuss selective permeability. Selective permeability. So being permeable means your ability to let things in. So if you're selectively permeable, that means you selectively let things in. So the selective permeability is provided by the hydrophobic chains of the lipid bilayer. Remember the fatty acid tails? Okay, those are the hydrophobic chains. And these are largely responsible for the selective permeability. Now, as it turns out, those hydrophobic tails are largely impermeable to most all molecules except for small nonpolar molecules. Now, if it's hydrophobic, that means it is nonpolar. And that kind of makes sense why maybe nonpolar molecules can get through. But for the most part, because of this, you can think of it as a wall where things cannot pass through. Okay? Because it's nonpolar. Most things are polar. But the way things get across the membrane is due to membrane proteins that are embedded within or on the phospholipid bilayer. And what they do is they help regulate the movement of ionic and polar substances. You may have seen sodium and potassium ion channels. But also small nonpolar substances may diffuse down their concentration gradient. Okay, so another membrane quality, we'll call this number 1. Number 2 would be it's self-sealing. That's good because if you were trying to build a fort around your city and it had any sort of, so it's selectively permeable, so you have a gate and you have certain ways that people can come in, be it their credentials or, you know, I guess if you want to completely destroy it, you can get a battering ram or something of that sort. You can destroy cells also like that, but the normal way would be going through some sort of process, like going through the gate. And the next is that it's self-sealing. What good is a wall if it's not sealed completely, right? And so this is a completely sealed structure, these membranes. And lastly, it's asymmetrical. It does not have a symmetrical element about it. And the asymmetry is actually very important. It says, your book says it's necessary for their function. So these membranes need to be asymmetrical, meaning they have random lipids and proteins, and cholesterol, you know, is a lipid. Proteins, these integral proteins, or these peripheral proteins and anchored proteins, and all these sort of things, these glycoproteins and phospholipids, all around it, making it asymmetrical. The lipid composition of each membrane is different, okay? And so you have many types of cells in your body. They all have DNA and organelles, but they differ in in various ways, okay? And the membrane, the phospholipid bilayer, is one way they might be different from one another. Membrane properties. First, we got to talk about membrane proteins. Most functions associated with the membrane require membrane proteins. So let's say you had this phospholipid bilayer. I'm just going to be lazy and draw it like that. There's your hydrophobic tails. And the question is, if it's selectively permeable and you have A that needs to get in, it's going to be blocked, right? But there's a way to get in, and we use membrane proteins most of the time. Okay, so membrane proteins are classified by either being peripheral or integral. And what peripheral proteins are is that they are kind of on the outside of a phospholipid bilayer, associated with the outside layer, but do not span the entire membrane, whereas integral proteins you see span the entire membrane. And so that's going to be the difference between the two. And each have their role. So what I encourage y'all to do is to watch this YouTube video detailing what these are. So you can have transport across the membrane. And so up here we have a phospholipid bilayer, and here's an integral protein right there. This particular drawing does not show any peripheral proteins, but you notice, like right here, you have ATP being used and a transport across the membrane. Maybe you, for those that have taken general biology, you recognize that to be active transport, because something got transported in by breaking off ATP, using that energy, yielding ADP and a phosphate. But now X has moved across the membrane because you used energy. You transported it, and it took ATP. That's going to be active transport, okay? There's also something called secondary active transport described there. Facilitated diffusion just means it goes down the concentration gradient, but it had to use a protein to get through. And of course, there's simple diffusion, which does the same thing as facilitated diffusion, except for it did not require a protein. X just hopped across that membrane down the concentration gradient, high to low concentration. So let's expound upon these concepts right here. Transport is considered to be a term that moves charged particles and polar molecules across the membrane. Notice I don't say nonpolar, and I don't say, uncharged. I guess they can be uncharged as long as they're polar, okay? But membrane transport is essential to all living organisms. Think about a country that didn't allow any import or export. It's not going to last too long, right? And so you have your body that's sending all kinds of things through your circulatory system, hormones, and your endocrine system and everything. And what happens is they need to go to a specific location and find a specific type of cell. And whenever it arrives at that cell, it needs a way to get inside the cell, because what good is products that are stuck behind a wall that's barricading the city? They need to get in, and your cell has various ways of letting things in or out, okay? So it's vital to living. If nothing's going in or out of the cell, if there's no communication, it's going to die, okay? It's going to not have necessary information to stay alive. It's not going to have the information known to keep other things alive. It won't be able to send things out of its cell, so on and so forth. So it's important for nutrient intake, waste excretion, gets rid of waste, right? You need to do that. And regulation of ion concentration. If you can't regulate that ion concentration, it's going to have ill effects, okay? So there's one type called passive transport. And what happens in passive transport is there is no energy input. Things just go down their concentration gradient. And then there's also something called active transport, which requires ATP because you're going against your concentration gradient. And I explained that right there. There's two types of passive transports. So you have active, which requires ATP, because you're going against the concentration gradient. And then you have passive, and there's two types: simple diffusion, and then you have facilitated transport or facilitated diffusion. And the simple diffusion doesn't require any protein, and the facilitated diffusion requires a protein to make sure it works. Okay, so let's see some examples of what I'm talking about. Well, actually, I'm not going to show you examples because your homework is going to make you learn these concepts, okay? So make sure you look at the homework, but I just wanted to let you know that active transport does require ATP. It is shown in this drawing on the upper right. And then, of course, simple diffusion is seen right here, where something just it goes across the membrane without a protein for help. And then facilitated diffusion requires a protein, but it's still going with the concentration gradient, that being high concentration going to low. Okay, now I just wanted to let you know that active transport goes against the concentration gradient, so it's going from low to high concentration, which is why it requires ATP in the first place. Think of tubing down a river, right? You don't require it doesn't require any energy to just flip down the river when the current is going in the direction you aim to go. But if you're trying to go up the current because you saw something that you wanted on the shore, it's going to require a lot of energy to go against the current, and it's the same kind of thing here. So without further ado, let's take a look at your homework. So I need you to describe what a sphingolipid is. I talked about that in the last lecture. And also what is a ganglioside and a cerebroside and sphingomyelin, and what each one is used for. I want you to dig into your textbook here and to really see what these things are. I want you to provide a reason for why oils are liquids at room temperature, yet fats are solids at room temperature, even though they are both triacylglycerols. And what I'm looking for is, one is typically saturated and the other is typically unsaturated. So chemically, what is going on? I want you to focus on the ability to stack and the ability to not stack, okay? And what that does to the melting point, and also blood clotting. I want you to, after watching that video described in the previous lecture, take a look at some of the structure of the eicosanoids, PGE2, TXA2, and LTC4, being prostaglandins and thromboxanes and leukotrienes, and determine why they are used in fatty acids, or why are they even in the fatty acids section in the first place. So take a look at the structure, see if it has a fatty acid, and what physiological effect each one causes in your body. What's the purpose of these eicosanoids, and how may it help you or hurt you? And what is a mixed terpenoid? I told you what a terpenoid is, and I told you what a terpene is, but what is a mixed terpenoid? And what I want you to do is find and give me 2 examples of mixed terpenoids from your textbook. I need you to look in your textbook. I'm really encouraging y'all to read this chapter. What's the major difference between LDL and VLDL other than the protein component? I don't want to know, one's higher density than the other. I'm looking for function. I'm looking for you to critically think. What is the difference and what are their purposes? What are they primarily used for? What's the difference? If you can tell me that, then I can see that you are understanding what's going on in this chapter, especially the lipoprotein part. And then tell me what the passive transports are and the active transport, and give me an example from your book of each. Sodium ion channels, for instance, and those sort of things. You may want to look at some videos. There's really neat facilitated diffusion examples where it kind of looks like here's your membrane, and then there's like a glove-looking thing right here, and then the ball fits in it, and then next thing you know, it kind of closes up, flips over, and now the ball's on that side, and then it lets it out on the other side of the membrane. And of course, active transport, what kind of processes require ATP to get in and out of the membrane because you're going against the concentration gradient. I want to point out, though, that these 2 I changed a little bit from the last slide, so from the last lecture video, so you may want to see that correction. And then what kind of foods are high in omega-3 fatty acids? Just so that you know, omega-3 fatty acids are really good for you. They're good for your heart health, cardiovascular health, and I just want to know if y'all can figure out what kind of foods are high in these, and maybe I can even encourage y'all to eat some more healthy foods. So all that said, I hope y'all enjoyed this lipid chapter. I'll be getting chapter 12 out to you soon enough. Have a great day. See what great love the Father has lavished on us, that we should be called children of God, and that is what we are. 1 John 3:1a.